¿Se escucha? ¿Y cuál es el pointer? ¿Y el pointer? Perfecto. Bueno, yo voy a dar una charla un poquito diferente. Behavioral informatics, I used to call this bioinformatics, but the physicist, the one present here, my husband, used to say, no, that's just for genomics, it's taken. I was like, okay, well, I am a biologist, I do bio. So I call it now behavioral informatics. And so I'm going to take you completely out of your equations and your systems and take you to healthcare. So it's going to be, pretty different from what you have been doing. So I'm going to give you two examples of how you can use modeling and particular machine learning for healthcare work I did at Psychogenics. I was the first employee of this company and worked for 16 years there doing drug development. And also what I did at Early Signal, this is a nonprofit I founded. We merged with another nonprofit. I worked there for 5 years. And then I quit, and so now I'm a consultant for both of them. So, what is behavior informatics? Is the application of modeling and machine learning techniques to analysis of behavioral data. So I'm going to tell you all that about networks, and that's it. I also have a big equation that represents everything I'm doing, and it's very complex. And I can tell you about this during the coffee break, and that's it. And so now let's get on. So what I did for Psychogenics is a company that's, it's a biotech and a contract research organization, so it works for big pharma, the bad people. And so what we do there, we try to bring the 21st century computation and signal processing tools to CNS drug discovery. So how does it work? I invented about 18 years ago some hardware. We developed the hardware with the Robotic Institute of Carnegie Mellon. And this, we call it SmartCube. And then we did a NeuroCube and PhenoCube. They do different things. You put mice inside. My first idea was I will do a little cube. We put it here on a mouse, and the mouse goes around and it transmits everything they do, and we understand everything that happens to them, but that didn't work. Electronics were too small, and so we had to put a big piece of electronics, put the mouse inside, and so this hardware challenges the mice in different ways. It doesn't let them go to sleep. So it creates a very rich behavioral data set. In this one, the mice or rats walk around and you just look at the paws from, oops, did I do that? Okay. So and then PhenoCube, that groups up to 8 mice, and we can track everyone, and we know who is who. There's also a chip implanted, so when they occlude each other and the computer vision cannot resolve the images, we can ask the chip, who's this mouse? So there's a lot on the hardware, and there's a lot of the computer vision. So in computer vision, there's now we are using completely different computer vision. 18 years ago, deep learning was not in fashion. So now we are changing all the computer vision to deep learning, which is really cool. And with computer vision, we know where the mice are, the shape, the posture, and we can also derive behaviors, what is that they are doing. All that data, so for example, SmartCube creates 500,000 points per mouse per session, is reduced to behaviors, and that's then used to do behavioral informatics. There's a law, that's a mistake. And so we can create different signatures. We can give a drug and see how the behavior changes, and we create a signature of a drug or a class of drugs. Or we can create a signature of a gene manipulation. So we have some knockout mice and transgenic mice, and we can see what's the difference of these mice versus their control mouse in any of these systems. So this is more for psychiatry, gait analysis, social and cognitive analysis. So this was protected with patents that we've been filing for many years, and publications describing the system. So this is a little bit what I already said to you. We go from 500,000 data points all the way to drug and disease signatures. The system in vivo can run more than 10,000 compounds a year, which for in vivo drug screening is really unheard of. And I will show you how successful this system has been. This is how it looks. This is SmartCube. It has a camera on the top and 2 cameras on the side. We have an observed behavior observed by the computer. The ground truth is what the human says. So we, in this case you don't see it, but we train each behavior creating ground truth with humans scoring the videos. And that's something I did for many hours 18 years ago. So now we are redoing the system with the new deep learning algorithms. This shows you just a little bit of the segmentation. So for those of you who don't do computer vision, the first thing you do is you find your object in your video, and then you do modeling of what the object is doing. So how do we do compound screening? So in the way pharma works, they say, well, we are interested in schizophrenia. Well, then what are the targets that are important for schizophrenia? Well, the dopamine D2 receptor, the 5-HT2A serotonin receptor. Let's do ligands that bind to this, and then we'll test them in animal models. So they choose the therapeutic indication. They need to have an idea of what target. They need medicinal chemists. They need a compound that will bind to that receptor. So they have a druggable target, a compound library that they can use for their experiments. They normally have a high throughput in vivo screen that tells you, yeah, this binds to D2, or it doesn't bind enough, or it binds but it gets off too fast. And they get their best compounds, and the medicinal chemists say, oh, I can do more of those, I can tweak them and make them better. And so eventually they get to lead compounds, and then you go to preclinical trials where you look for efficacy for schizophrenia, and then if it works and they are not toxic, you go to the clinic. And this takes at least 10 years, 20 years. I mean, it's a very slow and, especially here, very expensive process. The truth is that when the first antipsychotics were discovered, chlorpromazine was the first one, I think 51, and then there was a rush to get more antipsychotics. Clozapine is one of very effective antipsychotics. All of those were done without knowing why they work. They were done in a very different way. And then the molecular biologists came and said, oh, we know all these receptors, and now you can do it this way. This is going to be fantastic. Well, it's a total failure. There are very, very few drugs, especially for schizophrenia, depression, for psychiatric disorders that were discovered like this. I actually can't give you an example. So what we can do with the systems that I just described to you is go back to the origin. We can do what they did when they discovered chlorpromazine. So how does it work? Well, you need your compounds, but they may be for something else, maybe for erectile dysfunction. And now you say, well, these are good compounds. So I have my compound library. I'll put them in the in vitro system, in vivo system that I just described. And then we choose the therapeutic indication. Because I can now run drugs and say, these look like antidepressants. And I have no idea why, but they look good. So we choose a therapeutic indication there. We do have a candidate series, and we do improve the drugs with the medicinal chemists. We want to have drugs that stay there for long, that can be orally bioavailable. So we do this is the structure activity relationship analysis. And then the lead compounds go to preclinical, etc. So the idea is that when you get here, you already know that they work in vivo. When you get here, you still don't know if they work in vivo. So here you may have a lead compound, looks pretty, but it's a total failure very early. So you wasted all this time. And in here, this lead compound actually is already pretty good. So this is another way of looking at this, but I think that the important point is that there is a reference database with lots of drugs that are run and they are trained. So the algorithm is trained with reference compounds. So we tell the system, this is an antidepressant, this is an antipsychotic, this is an anxiolytic, and why we know that? Because they work in humans. So we train the system with drugs that we know what the effect is in human, and then we use a mouse to amplify that pharmacological effect. I'm not saying mice have a schizophrenia and I'm curing them, because that's really stupid. What I'm saying, it's an in vivo system with lots of very complex interactions of genes and pathways, and when I put a drug, I get an output that's useful. And I use that output to train an algorithm to recognize drugs, subclasses of drugs, like serotonin reuptake inhibitors, and classes like antidepressant, antipsychotics, etc. So once I have that database, I can put a novel compound, and I ask that train algorithm, what is this?
But the mice are healthy, or you have like models of certain diseases?
For this drug screening, mice are very healthy. And they're always the same. They've been the same for 18 years. The way the system challenges them is the same. The script has not changed in 18 years. But if you have a mouse model of, for example, a genetic disease, we have mice models of Huntington disease or autism, a number of mouse models, then you can create a disease-specific signature. And now you can treat, and I'll show you one example, you can treat a mutant, the mouse model, with a drug and see whether you can get rid of the signature. But a big database wouldn't be possible because breeding those mice is very expensive. So the drug screening is done with healthy mice. So